Okay, let's see if I can set up here. So tonight, I'd like to start off by acknowledging the traditional owners and echoing the call that we made before. One thing I'd like to add to that, because usually I don't get to be the first person introducing and making acknowledgements to country, but I'd like to add to that the fact that I'd like to honour our ancestors, or at least our Indigenous peoples of Australia, who are the oldest communities and scientists in the world, who've been passing on traditional knowledge, scientific knowledge, and some of the oldest cultures of transmitting that scientific knowledge, which some of us are only just recently tapping into now, but knowledge that they've had for thousands and thousands of years. So I'm going to talk a little bit about science, but more about the programs that I've started with the koala work. So to start off, I'm not a researcher who's looking at a big question of koalas. I'm not looking at the chlamydia questions. We have excellent researchers at QUT, University of Queensland, University of Sunshine Coast, who are looking at disease. They're looking at chlamydia research, whole genome research. Mine's actually looking at programs of outreach, programs of information, and programs that allow communities to become involved. And I think one really interesting thing and one really important thing is actually to present to people a way that they can get involved and a way that makes it rewarding for them to do so. And that's where I'm going to talk about tonight. Some of you may have seen me talk about the genetics of koalas and family trees and things like that. And that's essentially what we're looking at. We're starting off and focusing on the koala itself. Not the population, not the disease, but the individual koala. And by looking at the individual koala, the desire is to actually start to look at relationships, start to look at family trees, start to, once we collect enough data, ask questions that then lead to research. What's the sex ratio of the koala offspring? How many offspring does that female have over its lifetime? What are the types of the range that the koalas use? Where do the newborn koalas go that are not yet tagged and you can't track? And so that's kind of the area that I'm looking at, but it's going to take many years to develop databases to start looking at that. And that's what we've started now, is to generate databases that in the future could be used to either supplement survey work or to actually ask really interesting questions of the individuals, of the koalas themselves. I think when you get to things like surveys, for example, and Logan City Council is currently doing their survey for November, this work would not replace that, but it would complement that. It would complement in a way that the genetics might say, well, there's 50 koalas here, and when you do the survey, you might find 25. But from this work, you'd actually identify the koalas that you can't see. How's that possible? Well, when we do paternity testing of koalas, or if we do reverse paternity testing of koalas where you know you do not have the father, you've got 5 koalas, they all share the same father. Even though we can't genetically type it, we know that father is there. And babies keep getting born to that father, which means we know he's still breeding. That's the invisible questions that a database and large data sets could address. So that's kind of where we wanted to start. To get to the genetics and the science, which I'm not going to do too much tonight, is essentially we're looking at mitochondrial DNA to look at populations, simple sequence repeats, which is similar to the forensic science approach of paternity testing, relationship testing, YSTRs, much like your genetic genealogy, to identify the paternal line or the male line of inheritance. So we can essentially look at the genetic genealogy of koalas, and then SNPs as well, because it's works very well on degraded DNA. And all of this is being done on scats. So as we had, I think it was Councillor Tracy Davis, we also had Susan talk about scats and the wealth of information we get from scats. That's kind of where I'm focusing. And my expertise, apart from being forensic science and ancient DNA, is the ability to get that genetic information from the scats, right? That's the area that I quite am enthusiastic about. And to get started on this particular question of who's related to who and what are the relationships between different koalas and koalas within the population, Brad Lambert asked a question, can we do this from scats? Can we get the genetic information from scats that may allow us to do this type of question? So it really started with Brad's question that came from Education Queensland, it looked at the possibility of developing outreach programs with schools. That's where we started. So we got a little bit of money to build the DNA typing process. We're at the moment building the outreach program with the schools, because I really believe that if we're going to create, build and enthuse the next generation, we need to get them at the schools, right? And the school programs are doing brilliant jobs. Of course, the koala is iconic, so most certainly all the children who want to go into environmental science know about it. But what about the genetics, and what about the mathematicians, and what about the other youthful scientists, and what about the people who are not necessarily sure they want to go into science? And so we're building a program that will be delivered, or starting to be delivered next year, and it's going to go into grade 10. Why grade 10? Now grade 10 is a little bit later, but it's before they choose their senior years. But in grade 10 is when they actually have their genetics module. So while they're learning about genetics, genetic inheritance, some of you might know Mendelian inheritance, the laws of Mendel, where independent assortment and segregation and so forth, and the inheritance of genes, if we present that to school kids on koalas, and we present that to school kids with genetic markers from the koalas, that's very easy to understand, very easy to identify the inheritance patterns. And that's what forensic DNA typing does, and that's what our typing system does. So that students can look at that information and see exactly the inheritance patterns that are occurring, but we are enthusing those students into koalas, right? And so it's I think a very, very good way to start to expose that expose the students to that information. So grade 10 genetics unit is where it's going. It's at the Eco Centre here, which is where we're partnering with. We have other programs here at the Eco Centre. We're modelling it on that program, where we have students from schools all over Queensland come to the Eco Centre. It's usually 1 or 2 students from each school who come. They're usually selected by the school because they're like science-interested students, but it can be expanded further. And the idea for this program is that they'll come here, we'll talk about koalas, and take them for a walk to spot the koalas. Now, some of you were here early enough today where we walked down and saw 3 koalas, right? They're just at the bottom of the hill. They might be moving on now, but at the bottom of the hill we saw 3 koalas. And every day I walk through the forest, I can spot koalas, although I have to say, except one. I've lost my 100% identification. And so Monday this week, I couldn't find a koala, but every other day I do. And so we take the school kids through the forest, spot the koalas. Okay, which kid doesn't like to collect poo? Then we collect their poo. And it's even better when it's falling out of the trees, right? Fresh poo is even better. So then they will be out there collecting those scats. Then we take them to the laboratory and they extract the DNA from their scats. And they perform the DNA profiling. And they analyse the data to understand where they started, where the data finishes, and how the relationships can be built from that information. So this is starting with the Eco Centre. The funding for this started with Education Queensland. The funding has continued with the Department of Environment and Science with a Queensland Engaging Science grant to build up the DNA profiling systems and then to be able to deliver that, and we're starting that delivery next year. However, that model can also be applied to other schools. So the idea, as we move into next year, as we build that particular program, we can actually take it to other schools anywhere in Australia. And the part of that program is that we would go to the school and talk about genetics, talk about koalas, talk about DNA, and talk about poo, right? Then, if we can get the local schools enthusiastic about it, their environmental club and the other clubs within the schools, they will be able to then collect scats from their local koala population. We could go and introduce them to it, show them how to collect it, show them how to send it to us, but then we'd also show them how to analyse the data. And they would be analysing their koala population from a genealogical perspective and identify relationships and who's the mum and who's the dad and where's the baby and does he have a sister or a brother and so forth. So we're pretty excited with that program, and that's the first program that we're running with. The second program continues on from that as a citizen science program. And the citizen science program is not a far-fetched from the first one, except that we open it up to everybody else in Australia. And so in this instance, what we're hoping for is that everybody else in Australia, at least on the eastern seaboard, can start looking for scats and sending it to us. And so any population around Queensland, any koala population, we're hoping that enthusiastic individuals or community groups can start monitoring their koalas and their populations. They're already doing that, but collecting the scats and sending it to us. So this particular one is where, and we've got scats at the moment from Queensland, New South Wales and South Australia who are sending scats into us. At the same time, of course, right now, DNA typing of one single koala in South Australia is useless. But we've got to build that database. We've got to build the information so that it becomes more informative to us. And one of our community members who lives in the Lockyer Valley was very, very excited about being able to send scats from their local population, which is on their property, and sending them in. And they said, from all of them, as many times as I like, as many times as you like, even the baby ones. And so over time, that information, we might be typing the same koala 100 times, but over time, we'll be able to identify where that koala has been found. What range is that koala found in? Maybe where it's moving, right? So we can get all of that information from it. And the local individuals, and of course, many of us are interested in family histories and genealogies. And so we're kind of tapping into that human nature to understand relationships within the koalas. And that goes a lot further or way beyond population genetics. Right? So it gets right to the individual koalas. And we've got community groups from so far all over Queensland who have expressed interest, whether they be school groups or community koala care groups and so forth. The next program we've started is partnered with the Logan City Council, and in this particular case we received an Enviro grant from them to continue the optimisation of our DNA profiling system, because it's improving as we go. And so in this particular case, we're looking at, because this month is the Logan City Council's Koala Survey Month, and so we're tapping into that where individuals can send in their scats from that particular survey process. So this is just one of the partners that we've built, and the only reason I'm showing it is because they funded it, or parts of it. And so as we move forward, we'll be looking at koalas and their population and the relationships of those koalas to other populations around southeast Queensland and Brisbane. And depending on how much information we get, it might go way beyond that to the whole of Queensland, or to the whole of Queensland, New South Wales and Victoria and South Australia. So we'll kind of see how far that goes. So the key to this particular program for citizens is, of course, sending in the scats. And so for this particular program, we've actually got a replied postage paid free address for people to send them in. And so this is, of course, to facilitate the ease for the citizens and the communities to do this. So essentially all they have to do, and I've got some scats up here that some of you can look at if you want, but all they have to do is get their scat, fold it into a small piece of paper, right, or put it into a coin pouch or a coin envelope and send it to that address. What I also ask them to do is take a photograph of the koala. Now, I don't care if they take a photograph of the koala's butt, face, the tree, a fuzzy thing up there, or the scat on the ground. Because what I'm actually doing is I'm using that photograph to get the GPS coordinates, the metadata from the photo, not so much the photo itself. And so they email that into that email address, and I use that metadata to identify the GPS coordinates so that in the future we could look at range, migration and movement. So we're actually tying it to the landscape as well, which would supplement some of the monitoring programs. Of course, the sniffer dogs that I think Councillor Tracy Davis also mentioned, actually so did Susan, where you have a coordinated program to go and sniff out the scats in that particular territory, which is a great process for getting them quite rapidly. In this case, we're looking at subtle collection by the citizens, right? So they're going out, they're walking around, they'll just collect them as they go. It may take a long time for us to get data, but so far we've received hundreds of scats from around Australia. We're not keeping up with the profiling, but that's going to change. And so essentially we'll get a lot more data a lot faster than I expected. The challenge we have at the moment are fresh scats, so let's say the scats that are falling out of the trees, which some of my students thoroughly enjoy when I take them down to a tree that's raining scats, and I make them collect them because they're students, right? But essentially when we get down there and we collect the scats, within 24 hours we can get DNA very, very successfully and very reliably. Over time, as the koala scat gets older, it gets more and more difficult. And what we do instead, so I've got here, this is a nice scat, right? So this is one that's within 24 hours. And in this instance, we extract the DNA by rinsing off the cells on the outside. That's the epithelial cells of the koala. And then we extract the DNA from that, which is really, really easy to do. But when you get an older scat, like these ones here, right, these ones are over 3 to 4 days old. They start to dry out, and in this particular case, it's very difficult to get the DNA using the same method. But I don't give up, and so we go for grinding up the scat and extracting the DNA from the scat itself. And the amount of DNA that we're getting in comparison to, let's say, trace DNA at a forensic crime scene is about 20 times the amount, right? So we can solve a crime using 20 times less DNA. We can profile a koala from 3 7-day-old scats. And so we're developing and improving. When I say improving methodology, it's improving those processes as well. Now, there are researchers around that are looking at the microbiome of the gut of the koala, and that might be to capture the koala and collect samples. This, of course, is non-invasive, which some people have referred to as being beneficial for the koala community, so you're not causing any trauma to the koala. But, of course, getting the microbiome from the scats is possible. So we save all the scats that we extract our DNA from. So for future research, we're hoping to start looking at biomarkers and microbiomes and things like that. But that then goes into research. And today, I'm really here just talking about our programs. And so the programs that we've got, we're hoping will start to reach schools, communities, and other people across the country. Thank you.